Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Right before we jump into today's deep dive, please take a quick moment to hit that like button, subscribe, and share this with friends and family. That truly supports the Sand Science Experience, and it helps us keep bringing these targeted analyses directly to you. Today, we are undertaking a critical deep dive into one of life's greatest ongoing quality control battles, how our cells protect us from our most vulnerable genetic material, the mitochondrial DNA. We've synthesized sources that pinpoint the exact molecular mechanism regulating whether damaging, disease-causing mutations get passed down through generations.
Yeah, this is really a story about symbiosis, but a very precarious one. Life as we know it, eukaryotic life, it relies on this tight cooperation between the main nuclear DNA, the genome we usually talk about, and the mitochondrial DNA, or mtDNA. The critical problem you're looking at is the mutation rate. Because of where they reside and how they function, mtDNA mutates about 15 times faster than the nuclear genome. If the cell doesn't have a reliable, robust defense mechanism, that rapid mutation rate leads to genetic incompatibility, threatening the whole organism.
Okay, so let's start with the basics. The function of these organelles. We call mitochondria the cell's energy factories, right? Primary job is creating ATP through oxidative phosphorylation, OXPHOS.
That's correct. OXPHOS is, it's complex. It requires 5 massive enzyme complexes. We call them complex 1, complex 2, complex 3, complex 4, and complex 5. Together, they're built from over 90 different protein subunits. Crucially, while most of those subunits are encoded in the nuclear genome and imported, from the outside, 13 absolutely essential subunits are encoded directly by the maternally inherited mtDNA.
Only 13.
It's just 13. Think of it this way. The nuclear DNA builds the factory structure, maybe the walls and roof. But the mtDNA provides these 13 essential pieces of heavy machinery needed to actually run the power plant. If those 13 pieces break down, the whole factory just grinds to a halt.
And when damage happens to the mtDNA, it's not like an immediate shutdown, right? It's gradual. Which brings us to this concept, heteroplasmy.
Exactly. Heteroplasmy is just the state of having a mixture of different mtDNA types within one cell. Since every cell has hundreds, maybe thousands of mtDNA molecules, a new mutation initially only affects a small proportion. The situation is okay at first because the healthy or wild-type mtDNA can complement the function of the mutated ones. The cell can still function, sort of limp along, but that tolerance is also the danger because it lets these severe, potentially really bad mutations quietly survive, copy themselves, and spread through the cell population.
And if that keeps going, that 15x mutation rate you mentioned could lead to what the sources call a mutational meltdown over generations. We know nature has a kind of big picture defense against this, the genetic bottleneck.
Right, the bottleneck is our first line of defense. It happens during the development of the female germline, the eggs. Basically, the number of mtDNA molecules in those early germ cells gets drastically reduced, sometimes by 1,000-fold.
Wow.
Yeah. So if you start with a mix, heteroplasmy, and you suddenly reduce the total number that drastically, it accelerates the segregation of those different types. Imagine you have a big jar of mixed red and blue marbles and you just scoop out, say, 10. You're way more likely to get mostly red or mostly blue in that tiny sample.
Ah, okay. It forces the issue.
It forces the issue, exactly. It pushes the offspring towards either having mostly healthy wild-type mtDNA or a devastatingly high load of mutant DNA. This lets natural selection weed out the really bad cases at the level of the whole organism.
But the sources emphasize this isn't perfect, right? The bottleneck works well for really severe mutations. What about the milder ones?
That's the gap. That's the problem. Milder mutations can still slip through that bottleneck. They survive the big reduction event, and then they can accumulate over generations, eventually hitting levels high enough to cause disease later in life, or even take over completely. That's called homeoplasmy. So realizing this tells us that just relying on the bottleneck isn't enough. There must have evolved some kind of cellular quality control, a more precise molecular mechanism to police this transition, to get rid of the damaged goods before they're passed on.
Okay, this is where it gets really interesting. The main candidate for this cellular QC is something called mitophagy, which is basically the selective cleanup and recycling of damaged mitochondria using the cell's general autophagy pathway, right?
That's the one. And the breakthrough here is finding this specific enzyme whose normal job is actually to stop the cleanup crew. We're talking about ubiquitin-specific peptidase 30, or USP30.
USP30.
Yeah, USP30 is a Deubiquitinase, a DUB. It lives on the mitochondria, and its job is to take off these little tags called ubiquitin. Now, these ubiquitin tags are like the signal flag saying, hey, this mitochondrion is damaged, take it away, for removal via ubiquitin-mediated mitophagy, or UMM. So USP30 is effectively the brake pedal on the mitochondrial recycling system. It keeps the destruction process under control.
Okay, if USP30 is the brake, then well, if you want more cleanup, you need to cut the brake lines, right? Is that what they tested?
That's precisely the experiment they did. They used a mouse model that carries a specific mtDNA mutation, M.5024CT. It's structurally pretty similar to common human disease mutations, and they genetically knocked out USP30 in these mice.
And the result?
The result was clear. Getting rid of the brake pedal significantly enhanced purifying selection. Losing SP30 meant more ubiquitin tags stuck around, triggering UMM and allowing the cell to clear out more of the damaged mitochondria.
Do you have numbers on that? Like, how much difference did it make for the next generation?
Absolutely. The data was quite striking. Mothers who had 2 USP30 knockout alleles and started with a pretty high load, around 75% mutant mtDNA. They transmitted about 10% less mutant mtDNA to their offspring compared to mothers with normal USP30.
10% less. That's significant.
It is. That 10% difference confirmed that by removing the brake, USP30, you unleash this sort of latent, powerful quality control system that actively weeds out the mutant DNA.
This idea that the QC is just latent, waiting to be unleashed, that's really cool. But when does this selection actually happen? Is the mother's body doing this all the time, or is it timed?
The sources pinpoint the action to a very specific critical window. The maternal-zygotic transition, or MZT. Yeah, this is the period right after fertilization. Think of it like a mandatory factory reset for the brand new embryo. All the proteins and RNAs that the mother packed into the egg, they get rapidly degraded, cleared out, and they're replaced by new gene products made using instructions from both the maternal and the newly introduced paternal nuclear genomes.
So a complete system flush right at the beginning. Why is that timing so important for mitochondrial QC?
Well, because the father's nuclear genes are now in the mix, right? And the cell needs to make sure everything's compatible. If the maternal mtDNA is really faulty, it might not work well with the nuclear genes the father contributed. So by activating this QC check right at the MZT, the embryo does a final inspection. It makes sure the mitochondria it keeps are compatible with the combined nuclear blueprint before serious embryonic development kicks off.
That makes a lot of biological sense. Okay, but this brings us to the core puzzle, the paper tackles. If this powerful mechanism exists and USP30 is the brake, why do these damaging mutations ever reach disease levels in humans at all? Why isn't this cleanup running effectively when needed?
This is where the research solves a major paradox. It reveals this kind of negative feedback loop that the cell can get trapped in. Turns out, if a cell reaches a really high mutant burden, let's say over 60% heteroplasmy, that high load of faulty mitochondria actually starts to impair the cell's general ubiquitin proteasome system, the UPS.
The UPS, that's the cell's main system for breaking down and recycling proteins generally, right? Like the main garbage disposal.
Exactly, the general garbage disposal. And when it gets overwhelmed by all the trash coming from the faulty mitochondria, it starts to break down globally. This leads to a reduced rate of degradation for all proteins marked with ubiquitin. But crucially, the sources show this specifically hits mitochondrial ubiquitination hardest. It selectively impairs the targeted cleanup mitophagy, while general autophagy might still be chugging along, more or less.
Wait, hang on. So the damage caused by the bad mtDNA actually sabotages the very system designed to get rid of it.
That's right. It's a kind of terrible biological irony.
Wow, that explains a lot. It's like the problem protects itself once it gets bad enough.
It does. And this really helps answer a long-standing scientific question around the maintenance of wild-type hypothesis. For years, people thought maybe high heteroplasmy cells kept functioning by just making more mitochondria, trying to synthesize more healthy DNA to compensate for the bad stuff.
Right, they thought the cell was trying to build its way out of the problem, build more factories.
But these sources reveal it's actually the opposite. The reason you see more mitochondria and mtDNA structures, these nucleoids, accumulating in high heteroplasmy cells, it's because of decreased destruction, not increased synthesis. The cell isn't ramping up production. It's basically shutting down the protein degradation pathways. It stops throwing things out. This is probably, a desperate short-term survival move to hold on to whatever functional wild-type DNA it has left. But the terrible consequence is that it allows the highly mutant mtDNA to survive and get passed on. It's a short-term patch that leads to long-term disaster.
That completely reframes how we think about mitochondrial disease progression. It's not a failure of making stuff, it's a failure of selective recycling. And that failure points right back to a clear therapeutic target, that inhibitor, USP30.
Absolutely. USP30 is now a really strong therapeutic candidate. And the research shows you don't necessarily need to do genetic knockouts like in the mice. You can effectively restore that cleanup, the UMM, by using small molecule inhibitors. They used one called compound 39 or CMPD39.
Compound 39. Okay, so what happened when they treated cells with this drug?
Well, they tested it in cells that were forced to rely only on OX-PHOS for energy. They did this by growing them in galactose instead of glucose, making them really depend on their mitochondria. And they saw two really powerful effects. First, CMPD39 preferentially reduced the overall level of mutant mtDNA, the heteroplasmy, by about 3 to 5%.
Okay, a noticeable reduction.
Yeah, but maybe even more importantly, it significantly decreased the number of cells that had that really high dangerous level of mutation, the ones above 60% mutant load. That population dropped by a full 10%.
10% fewer of the worst affected cells. That's promising.
It really is. It proves you can chemically unlock the cell's own latent QC mechanism and selectively push it to get rid of the most damaged mitochondria.
The implications here are huge.
Yeah.
This could potentially offer treatments, maybe even preventative strategies for rare inherited mtDNA diseases. Those affect about 1 in 8,000 people, right?
That's the number, yes. And think back to when this selection happens most strongly, that MZ2 window. This opens up the possibility, maybe down the line, for a truly transformative intervention. Could we potentially treat early embryos after in vitro fertilization using an inhibitor like this, actively reduce the mutation load they inherit before implantation, essentially correcting the problem before development even properly starts?
That is exactly the kind of next-level genetic intervention that starts to feel like science fiction becoming reality.
And, don't just think about the rare inherited diseases. There's growing evidence that these heteroplasmic mtDNA mutations build up over time. They seem to contribute significantly to common late-onset problems like Parkinson's disease, certain cancers, and just general aging.
Really, so it's broader than inherited conditions.
Potentially much broader. If we could inhibit USP30 safely and effectively, maybe boost mitochondrial quality control throughout life, the potential benefits could extend far beyond fertility treatments and rare diseases. It could impact aging itself.
What an incredible deep dive.
Yeah.
We started with this fundamental threat, the rapid mutation of mtDNA, and we ended up pinpointing USP30 as this key enzyme, basically a brake on the cell's quality control. And the exciting breakthrough is that by chemically releasing that brake, maybe with compounds like CMPD39, we can unleash the cell's own cleanup crew. This could drastically reduce the inheritance of damaging mtDNA, potentially preventing disease right at the start of life, and maybe, just maybe, even pushing back against some aspects of aging later on. We really hope you enjoyed this journey into molecular biology and genetic medicine. Thank you for joining us. Please take just a moment to like, subscribe, and share this information with anyone curious about the future of genetics and how our bodies work. We appreciate you being part of the science experience.